In our lab, we are mainly focused on understanding the link between chromatin and human disease. As you know, every cell type of our body has the same DNA content, so the DNA is exactly the same, and yet every cell is different. So the genome, the information is the same, but the way in which the genes are expressed in different cells is different. For example, in our hematopoietic cells, we do express genes like hemoglobin, but we don't express genes like, for example, which are typical for neurons. So the specific expression of genes in the different cell type is governed by a complex, multi-protein complex that regulate exactly the chromatin structure. That's the way in which the DNA is packed into the nucleus. Among the different protein complexes, in the last 3 years, we have been focused our research in understanding the role of polycomb group of protein, which is a very interesting set of protein. And then studying these proteins, we highlight a link between chromatin, gene expression, and human disease.
As Luciano was explaining, polycomb is a team of proteins that work together to mediate its function, and its function is to repress gene expression. So polycomb is indispensable to repress genes that should not be expressed in a concrete cell type. One of the main subunits of this complex is the protein that is called Mel18. In the lab, we have unveiled the role of Mel18 during ESL differentiation. In particular, we have seen that Mel18 is essential for the differentiation of ESLs toward cardiomyocyte cells. If the cells do not have this polycomb component, the cells they cannot beat as a real heart.
Since the availability of the genome in the last 15 years, the genome of the humans, and mouse and other species, we can study multiple genes at once. In order to study what Alexandra and Luciano was explaining about the protein of the polycomb group Mel18, what we do is to do bioinformatics analysis. To extract important information from this huge volume of data, we need two things. Computers to process this data, because people cannot process this data manually, and bioinformaticians that learn how to use these computers and process this data and interpret the results to get new conclusions that can help to progress in the lab with new experiments.
So to conclude, the experiment that we perform in the lab and the bioinformatic analysis that Enrique was mentioning allow us to understand the role of chromatin in different type of human disease. For example, what Alexandra referred before, Mel18 is important for differentiation of cardiac cells. And what we found is that deletion of Mel18 is present in different type of human disease, including cardiopathies. In the lab as well, we are studying, we don't have time to talk about that, but we also study other type of human disease, including leukemia, prostate cancer, and breast cancer.